Hello dear students, this is a special session. Here I would like to give you some information regarding the control of the cell cycle or control of the cell division, because we are going to understand a very important concept of the human health and disease chapter, that is cancer. So we know the definition of cancer, it is nothing but uncontrolled mitotic cell division results in the formation of tumor, correct? But we need to understand why this tumor occurs. Okay, why the cell undergoes cell division which is uncontrolled. Okay, but if you take a normal person, why the cells are like undergoing cell division in a controlled manner? There are factors actually which controls the cell division. Okay, so in the first episode when I was discussing about cell cycle, I said like I'm going to explain it in depth about the cell cycle and its control in the next year, that is actually in the concept cancer. So we will explain that. Please note down each and every point, very important actually. The first question is a very simple question: why cell or cells divide? In the initiation phase of our life, that is actually the formation of embryo, yes, obviously we need to increase the number of cells, correct? Okay, that is actually in the first phase of our life. Why? To increase the number of cells. Okay, so that is actually the important thing, increase the number of cells in our body. Yes, that is nothing but growth itself, for growth. Second thing is to replace the damaged, I'll just write damaged cells, or maybe to replace the lost cells. Okay, so this can happen anytime in our life actually. Maybe due to a small injury or accidents, or maybe a normal death of the cells, that is apoptosis. Okay, to replace this lost or damaged cells, we need the cell division process. Okay, so we just write one more, that is to replace the lost cells. Hope you can hear my voice. Okay, I am taking class in the lab, bio lab. The next is, okay, so we just write how cell cycle. Hope you understood. Cell cycle in the sense it has two important phases. One is interphase, the other one is M phase. In interphase, you three small subphases. They are G1 phase, S phase, G2 phase. Then comes the M phase. In that four phases are there. You know that: prophase, metaphase, anaphase and telophase. Correct? Okay. After the completion of this M phase, that is telophase, the last phase of M phase, the cell enters into a kind of static phase, that is called as G0 phase. Okay. In the G0 phase, actually the cells decide whether to continue further cell division or stop, like that. Okay, as it is. So this is the thing. Okay, how cell cycle is initiated. So this is applicable to this and this. Okay, hope you understood. We'll write here how cell cycle is initiated. First I'll write all this cell cycle, that is the division of the cells, is actually initiated by certain factors. Okay, that is nothing but called as cell signaling pathway. Okay, if one cell actually decides, okay, it requires more neighbors, then obviously it's going to send some proteins as a messenger to initiate cell division. That's it. Okay, so we'll see that the first one is actually called as the autocrine signaling, cell signaling. The second one, let me just write here, that is paracrine. Auto means self, para means from other cell. The third one is endocrine. So these three signaling pathways actually initiate cell division or cell cycle. Okay, so we just go with the autocrine. Suppose we take a cell from a human body. So this means, okay, so we will take this human body as an adolescent human, like 16-17-year-old fellow like you. Okay, that means the growth phase is already completed, correct? Okay, he is not in the initiative phase of his life. We just take one cell from his body, any type of cell, any cell, you take any cell. Okay, we have taken one cell, we just draw it here, and we know it has got nucleus and the genetic material, and it has got cell organelles, everything is there. Now what is this autocrine? Okay, Now let us imagine that very close to this cell there are other cells. Other cells, we just take one more cell, the neighbor cell of this cell. Okay, so it has also got its genetic material, everything, normal cell. But this cell is now damaged. Okay, what I am going to do is I'll just erase this. That means damaged, lost, something has happened. Now we have to replace this lost cell. How exactly it is initiated? That is nothing but by a process called as cell division itself, right? Okay, but we need somebody to initiate that. Who is going to initiate that? It could be the cell. Okay, we just imagine. So this cell is the neighbor cell of the same tissue. So the same cell should be produced. Correct? Okay, the same type of cell, because you know group of similar cells makes tissue, right? Let me just talk about the autocrine. Now this cell, to replace that lost cell, which is going to produce obviously protein. Okay, it's going to produce a type of protein. There are millions of proteins actually. Okay, we just consider protein alpha. Protein alpha is now produced, and this protein alpha is produced because of transcription and translation process of the genetic material of this cell. Yes, we know that. And this protein alpha now again actually comes into contact with the same cell membrane here. So this protein alpha now acts like a ligand that is going to bind to the receptor. Cell receptors will be there on the cell membrane of the same cell, or it could be other cell, but in this case autocrine, and it's going to bind here, and now it initiates cell signaling pathway. Okay, in the last year, first FUC have studied about cell signaling pathway, a little bit about endocrine, that is how exactly hormone action is controlled, mechanism of the hormone. Okay, so same, it now actually enters into the cytoplasm and now binds to the genetic material. Now this genetic material will produce some other proteins. Okay, so this protein again binds to the same genetic material, and now cell division is initiated. Cell division is now initiated of the same cell. Got it? So this is autocrine. If the same cell initiates its cell division, its own cell division, then it is autocrine signaling. Okay, hope you understood. Now it undergoes cell division and produces two cells. Yes. So the original cell is retained and the lost cell is replaced. Done. So this is autocrine. Okay. It is normal process. It is happening in our life, in our body actually. Next is paracrine. Okay, paracrine is very simple now. What I am going to do is I am going to take one more cell. Okay, let us say pancreatic cell. This is pancreatic cell now. Pancreas, it has got its genetic material now. Okay, so here I am going to take the intestinal cell here. I will take one more cell, that is intestine. Now this cell is now lost, gone, damaged. Something has happened to this cell, damaged. I want to replace it, but autocrine signaling is failed in this particular case. So it has to go with another signaling, that is paracrine. That is the neighbor cell. What is the neighbor cell now? The pancreatic cell is the neighbor cell of the intestinal cell, correct? Yes, belongs to different tissues. Both are entirely different cells. It is going to produce a protein that is called as the beta, for example, actually. Okay, and this beta is going to bind to the neighbor cells. Like let us say one pancreatic cell which is already now damaged, lost. I want to replace this cell. Let me just take one more intestinal cell that is actually the neighbor cell of the same intestinal cell itself. Okay, it's going to bind to this. The beta will bind to this, and again the same action is initiated. That is nothing but cell division. Got it? If the cell division is initiated from another cell, okay, which is not a neighbor cell or like another cell, then it is called as the paracrine. Got it? It could be neighbor cell also can initiate, or it could be faraway cells can also initiate cell division. That is paracrine. One thing to remember, that the cell division is initiated from other cells, not the same cell. If it is by the same cell, then it is autocrine. Got it? Okay. What is endocrine? Endocrine, we all know that hormones. Yes, endocrine glands. So let us imagine that a type of hormone, any hormone, you imagine pituitary, any hormone, okay, or you can imagine a hypothalamus hormone, actually any hormone. So that hormone is now released into the blood stream and finally reaches the targeted cells, and then it binds to this targeted cell and initiate this cell to undergo cell division. That's it. Cell division is initiated, done. So it is going to produce two cells. This is actually a common process or mechanisms which is occurring in our body every day. Got it? Majority autocrine signaling will be there. If autocrine fails, then it goes for paracrine. If paracrine fails or requires more effective signaling pathway, then it goes for endocrine, like that. Got it? Hope you understood that. This is actually the thing I explained how exactly cell cycle is initiated. initiated. How exactly cell division is initiated? These are all normal process. But, okay, we just erase this. What if, okay, so the cell now follows autocrine, paracrine or endocrine pathways, fine, chanta hai. But what if the cell division is uncontrolled? Yes, we all know that if you take human cell, okay, so this is human cell and which is undergoing cell division. So cell cycle in human, which includes interphase as well as M phase, takes 24 hours, correct? In human cells, note this point. 23 hours for interphase. If you want, you can note this point. 1 hour for M phase. So total 24 hours, and ultimately 2 new daughter cells are produced, that is mitosis. Okay, and this is actually the exact number of cells that has to be produced within the given time. Yes, that is normal. So this is now controlled process. Control, everything is under control. Okay, that's what our body shape is normal. Okay, we are not suffering from cancer. What if, okay, so let us just imagine that these two cells, daughter cells which are produced, should undergo or enter into G0 phase. After M phase, G0 comes, that is actually kind of resting phase. Here the cells will undergo some signaling pathway like autocrine, paracrine or endocrine, whatever it is, then decides whether to go for further cell division or stop here itself. Got it? This is controlled process. Now, in the case of cancerous human beings, the two cells which is now in G0 phase will not listen to any factors because they are actually kind of like autocrine activated. It's like the self-activated. They will not listen to any of the body factors. They will not listen to this control mechanism. Okay, they are on their own way actually. Okay, so now they undergo further cell division. See, in the next day, that is actually in the 48 hours, again you get 4 cells. This shouldn't happen, but happened. Got it? It should not take place, but it has taken place actually. This is the thing. So this is uncontrolled. Got it? So this is now leads to tumor formation. Tumor formation is nothing but cancer, early stage of the cancer. Hope you got it actually. Got it? So this is the thing. In our body, okay, so we just erase this. This is now uncontrolled. So it is understood that the controlled or uncontrolled cell division normally takes place within the cell cycle steps. It could be in interphase, yes, G1 phase, S phase or G2 phase, or it could be in prophase, metaphase, anaphase or telophase, or in G0 phase. It could occur anywhere. Got it? So this is the thing. And moreover, everything is controlled by our genetic material, that is the DNA. Yes, you take a cell, young cell. Now this cell is undergoing cell division in maybe 1 hour. Got it? It's now young cell now. It's like young cell in the sense like in the initiative phase of our life, or it could be like in the second phase, that is to replace the damaged or lost cell, whatever it is. Okay. So this is now the DNA. So DNA will be there here. And let us assume that it is now entered into the first phase of the cell cycle, that is G1. What's going to happen to this now? It's going to increase its size and it's going to increase its metabolites like the chemicals. Why? It's nothing but preparatory phase, correct? We know that. The next phase we say that okay, finally the cell has grown and it has become very big now. It is ready to enter into the next phase, that is actually called as S phase. Alright, here. In the S phase, what are you going to see is replication of the genetic material, that is the DNA length increases. Got it? 2C becomes 4C. The chromatin now length increases to the double fold. This is it? Yes? Okay. Next is, why this happens? Because the cell has to divide to produce 2 daughter cells. So that means that we actually know the one piece of the DNA goes to new cell, the other goes to the other cell, other daughter cell. That's it. Very simple. The next phase is actually the G2 phase. Okay, here we know that some proteins will be synthesized. Okay, why proteins will be synthesized? For future. That's it. Okay, it has to be segregated equally into the daughter cells actually. So all these things you know that. I will not explain much about it. This is actually the normal pathway of the cell cycle. But here, let us say this is actually in the G0 phase. Okay, now the cell is entering into G1 phase. Somebody should be there to initiate this cell to enter into the G1 phase. Who is that somebody? There are factors actually. This is nothing but the controlling of the cell cycle. Okay, cell cycle is controlled by two irreversible points. Okay, we just write here, somewhere else here. Cell cycle is controlled by two irreversible. It cannot be reversible. That's it. Okay, once done, done, gone. You cannot do anything much about it. Got it? Irreversible points. That is the first one. Let me just write the first irreversible point, that is replication. One is replication. Once replication is done, in this case, yes, in the S phase, you cannot reverse this mechanism. You cannot go back to this phase. Once done, done, end. Got it? That is replication. At the same time, another checkpoint, or irreversible point after replication is, if after G2 phase the cell will enter M phase. Okay, forget about the metaphase, anaphase. In the anaphase, what happens? Segregation, separation of the chromatids occurs, correct? The sister chromatids should separate. Okay, so once it is separated, you cannot rejoin them. Done. So, got it? So, that is separation. I think I'll write here, hope it is visible to you. Separation of sister chromatids. Okay, separation of the chromatids in the anaphase. These two are the first irreversible points in the cell cycle. Next is the checkpoints. Okay, we just go with this checkpoints. So what is this checkpoints? These cells are egg cells, they do not know their fate. Okay, fine. So now let me just tell you about the checkpoints, which is also irreversible in cell cycle. The checkpoint is G1 phase, the first checkpoint. The second one is S phase, the second checkpoint. The third one is G2 phase, the third checkpoint. So in cell cycle you have three checkpoints. What is this checkpoint? For example, if this cell, which is now in G0 phase, now decides, okay, let me just enter into cell cycle now. Okay, so now it enters into the cell cycle. Okay, when it enters into the cell cycle for its cell division, let us say due to some factors, let us say mutagen. Okay, what is mutagen? Mutagen is nothing but the agent, anything. It could be ultraviolet radiation, UVB rays, gamma rays, whatever it is, or chemicals, which now initiates uncontrolled cell division, or which now damages this DNA. Okay, so this is the normal DNA, and now it is being damaged. Okay, now it is damaged. Some base pairs or some nucleotides are now damaged. Got it? So this is the thing. Now some bases are now removed, deleted. That is actually the mutagen which causes mutation in the genetic material. But it is already now entered, jumped into the cell cycle now. It cannot come back. Got it? Now we need somebody to repair that or throw that cell away from the cell cycle. This is very important. Otherwise, if it is not controlled in these checkpoints, obviously we will suffer from cancer. This is the thing. Okay. We every day actually in our life, every day the genetic material is being damaged. Got it? But we have a wonderful system in our body, that is checkpoints, three checkpoints. It is going to repair the damaged DNA, or it is going to stop or kill the cell itself. Okay, to prevent the further loss or damage, that is suffering from cancer. That's it. Okay, G1 phase. In the G1 phase, there is some factors. Let me just write that factor. That factor is called as CDK. Please note this point. Very, very important. You may get this for your competitive examination. In the G1 phase or S phase or G2 phase, there is a kind of enzyme which now controls the cell cycle. Which controls the cell cycle. That is CDK. What is the full form of CDK? Let me just write here. Cyclin. Cyclin is a protein which is synthesized by these cells. Whether a normal cell also can synthesize cyclin, or the DNA damaged cell can also synthesize cyclin. A protein. It's a protein. Cyclin dependent. Kinase. Okay, so we just write kinase. Hope you can see this. Cyclin dependent kinase, that is the full form of CDK. If you take MBBS, obviously we are going to study about 4 different types of CDKs. CDK1, CDK2, CDK3, CDK4. But for POC level, it is enough. Just imagine CDK. There are 4 different types of CDKs, which normally they are very specific to particular checkpoint. Between G0, G1, there is one CDK. called as CDK2. Okay. Then between G1 to S phase there is another type of CDK, cyclin dependent kinase. Okay. Then between S to G2 again one more is there, like that. Fine. This is actually one important factor which controls the cell cycle. What is the main function of this? It is going to stop the damaged DNA cell. Okay, the cell which is not damaged, that is DNA is damaged, so it's going to stop from entering this cell into the G1 phase or S phase, like that. That is the main function of CDK actually. If it is not damaged, obviously they are going to allow the cells to enter into cell cycle. That is the main function of cyclin dependent kinase. It's very important, which is very important for this entire cell cycle process, for the success of the cell division. CDK, remember this, that is number one. The other important factor is MPF. What is this MPF? Another factor which also plays in cell cycle, that is maturation promoter factor. Okay, we just write here, hope you can see this, maturation promoter factor. Not necessary, you are going to study in your higher classes in MBBS actually. Okay, CDK and MPF, these are the two important factors which plays an important role in the regulation of the cell cycle. If they are not there, obviously, every day, every person, each person will suffer from cancer. That's it. Okay, this is actually a simple way to understand. They help in the regulation. Okay, so now why this CDK actually important in the control of the cell cycle? That is, each cell, as I said earlier, going to produce a protein called as cyclin. Got it? This cyclin is, whenever actually, it acts like, what do you say, you have already understood in Lac-Operon concept, right? Lac-Operon concept. Something is produced, okay, which is going to always binds to the promoter region and blocks the transcription process. Correct? The same way, the cyclin also goes and binds to the some promoter region of the DNA and blocks the transcription process. If the transcription process is blocked, obviously the cell will not enter into this stage, the further stages. So we need somebody to prevent cyclin from entering in or blocking the promoter region. So we need CDK. Cyclin dependent kinase will bind to this cyclin and blocks this cyclin, and as a result transcription process will be initiated so that you see the further cell cycle events. That's it. So this is the thing. Okay. So this is important. Next is, let me just try to write one more thing. The third important factor is P53 gene. So what is this P53 gene? Note this point. P53 gene is also called as tumor suppressor gene. Note this point. Very important. Tumor suppressor gene. Tumor suppressor gene is nothing but called as P53, recently discovered gene actually. Where exactly you see this P53? This P53 gene is situated in each cell of our human body or our body. Got it? So this is the thing. P53 is seen everywhere. Okay, in the genetic material of human cells. Done. What is the main function of this P53? The P53 acts like a doctor. Note this one, very simple. Doctor, if the DNA of the cell is damaged, which is now undergoing cell cycle, which is going to inspect that entire DNA. P53 means it is nothing but a gene which is going to produce the protein. The protein will inspect. That's it. Simple way actually. Okay, but let us understand in the easy way. The P53 gene will inspect or the product of the P53 will inspect the entire length of the DNA and checks whether or kind of it diagnoses whether the DNA is damaged or not, if the DNA is mutated or not. If it is mutated, it's going to block the entire process, that is cell cycle. If the DNA is not damaged and it is normal, it's working normally, then it will allow the cell to enter into the cell cycle. Hope you understood? Three factors: CDK, that will bind to the cyclin, allows the cell to enter into cell cycle, and another one is maturation promoting factor, which allows the cell to grow, maturation. Got it? So that is actually the thing. Where exactly you see the high concentration of MPF? Okay, so we just write there. Here, exactly, MPF concentration will be very high in the G2 phase. If you want, you can write down. Otherwise, they are normally there, but the concentration is high in this case because G2 is now entering into the M phase, that is prophase, the first stage of the M phase. So So MPF is required for it. Done. P53 acts like a doctor, which is also called as tumor suppressor gene. Note this point, very important. Which is going to inspect the entire length of the DNA and checks whether the DNA is damaged or not. If it is damaged, it's going to kill the cell itself, there itself, so that the person will not suffer from cancer. Okay. What if the gene P53 is itself mutated? The doctor is itself suffering from cancer or suffering from a disease, so he cannot diagnose. That's it, done. So P53 gene is a kind of boon, at the same time it's a curse also. If something happens to P53, obviously the person will suffer from the cancer, because checkpoint is not perfect now because of the mutation in the P53 itself. Got it? So note this point. So if something happens to CDK also, same thing happens. So cell will undergo uncontrolled cell division. That's it, gone like that. Okay, there are so many things actually, will not go in depth. Fine. So this is actually the thing you need to know before cancer. So ultimately, what is cancer? Cancer is uncontrolled mitotic cell division. Got it? Okay. Now I would like to add one more point here. I forgot in the first step you see when I was taking the cell cycle concept, that is APC. What is this APC? APC is nothing but anaphase promoting complex. Okay, so anaphase promoting complex. Okay, that is actually APC. Note this point. Now, what I'm going to do is I'm going to draw a chromosome. Note one more point. Where exactly you see highly condensed chromatin? Where exactly you see highly condensed coiled chromatin? In the metaphase. In CET or NEET could be asked. In metaphase you see highly condensed, tightly coiled structure of the chromatin will be seen only in metaphase. Very important. Note this point. Okay, I am going to draw a chromosome. So this is now one sister chromatid and this is now another sister chromatid. They are joined by a structure which is called as centromere. The question is now, so far I have not explained about centromere. How many centromeres are there? Okay, so we just draw only one chromatid. So one chromatid always contains one centromere. The other sister chromatid also contains its own centromere, but they are going to join, correct? So that is actually normally seen in the metaphase. Actually you see X-shaped structure. That means here you see two centromeres. Okay, two centromeres will be there in one chromosome. Okay, but two chromatids will be there. Fine. These two centromeres here, exactly the junction between this centromere and this centromere of two different sister chromatid, you see a protein. I think I better use a blue color here. There is a protein actually which acts like a gum. They actually adhere these two centromere so that two sister chromatids can join and make one chromosome. Name that protein which joins two centromeres or which joins two sister chromatid. That is nothing but called as cohesive. You call it as cohesive. Again note this point. Cohesive. Got it? Cohesive. Now the thing is, in the anaphase stage of the M phase, what is the important step you see? The important step is separation of these sister chromatids, correct? Obviously, we know that. That is actually allows the equal distribution of the chromosomes between or in the two daughter cells, new daughter cells actually. That's it, right? So for that, we need to break this cohesion. So we need a protein. That protein is called as APC, anaphase promoting complex. This is going to produce one protein actually, that is saparin. Okay, it's not required for your level. So APC will produce a protein called as saparin. This saparin will come and bind to the cohesin and now it destroys this protein, that is cohesin. Once the cohesin is destroyed, that is gum is lost, so they can easily separate. Done. So note this point. What is the function of APC? They help in the separation of two sister chromatids in the anaphase. So this is actually important thing. Hope you understood all about cell cycle and cell division. How exactly cell is controlled, cell cycle is controlled, or cell division is controlled. If it is not controlled in the checkpoints, obviously what's going to happen? The thing is nothing but uncontrolled cell division, which leads to uncontrolled or kind of like infinite number of or infinite increase in the number of cells. That is nothing but called as cancer, tumor. Got it? So that's all about from this session. Hope you understood.